Good morning. All right, we'll get back with the second half, which will be, first of all, Robert Weatheritt talking on Beyond Oil and Water. At first I thought it was a vinaigrette recipe, and then I remembered that was vinegar. He's going to talk some exciting new work that he's involved with, but also in the whole field of a lot of molecular biology of well, I'll leave it to him. And then I'll follow that, and then we'll have a question and answer session where you'll be able to ask your question. So thank you very much, Robert.
All right. Thank you very much, Anthony, for the kind introduction. Please. Even before I began. So today, me and Anthony's talk is now going to delve into the inner workings of the cell more and more. So what I'm going to try and talk to you and explain to you this morning is a new concept and some new ideas in molecular biology, which we're trying to use to understand how the cells, the inner workings of the cell, and how this leads and contributes, seems to be able to contribute to the pathology of neurodegenerative diseases. So the paradigm of how, and this is particularly associated with dynamic compartments and compartments within the cell. So the traditional way of thinking about compartments is in terms of for something many of you have probably seen, for example, when cooking. When you mix oil and water, they don't mix together, they partition into two separate elements often. An example of this here, where you find these small oil droplets forming. Another example is, for example, when you get a bilayer. And some of the details and more details of what's the molecular details of these the droplets are here. The classic one, for example, is the monolayer, where you see a monolayer of oil forming on top of the water. But more interesting in terms of biology is these so-called vesicles. In essence, these vesicles are these kind of oil droplets when you have a small drop of water actually stuck within the oil droplets. And this is interesting because when the environment around this vesicle can change dramatically, and yet the environment within the vesicle, within the water droplet within the vesicle, actually stays very, very stable. And this is massively beneficial for the basic biochemistry of life, because you have a stable environment for which to survive. And as humans, we're very familiar with the importance of a stable environment. And, for example, particularly in our houses, for example, the inside of our houses is very well maintained in our buildings. Outside, it can be minus 20 or plus 50, it can be raining or snowing. But within our buildings, within the buildings we make, we have a very stable environment which allows incredible productivity both in work and as well as family lives. And we're not suffering with the, trying to deal with the cold. We have a stable environment inside. And this is bricks. This is basically due to the permanent buildings we build, the brick and mortar which we have around us. And in the same way, the cell has a very similar mechanism for creating compartments which create specialized functions. And this is just a kind of classic picture of a cell. And for example, in the cell, you can have the nucleus, which is like the library of the cell. You have the endoplasmic reticulum, which are like the factories of the cell. You have the mitochondria, which are like the power stations of the cell. All these compartments, all these features are very important for and are semi-permanent features of the cell. However, this kind of semi-permanence comes with a penalty, of course. For example, if I want to get out of a building, I can't just walk through the walls. I have to find my way out, so I need to go up the corridor, up some stairs, and out the door. And of course, this takes time. In a very similar way, if I want to get rid of this building and build something else or have another feature here, it's quite an extreme thing to do. I'd have to knock down the whole building and start again. And obviously, this takes time, and this takes penalties. And there's obviously, though there's some features which a cell wants, which are permanent, there's a whole host of other things which it only wants for a few instants or in a temporary way. And this is what the cell seems to do. And so rather than having something which is permanent and difficult to get out of, we want something which is temporary, which you can get out of almost through the walls, and you can put up and down and replace. So in essence, in architectural terms, I'm basically talking about a tent, in essence. So as you can see here, tents are very easy on the whole to get out of. And also you can put the tents up and down, and you can store them very, very simply as well. I could watch this all day, I'm not going to lie. So I'm going to move on. So in the cell, very similar things happen. So as you can see here, this is just a video of a protocell, and the purple is just the inner membrane of the cell. And as you can see over time, this cell has the ability, if I just fast forward a little bit, so at the moment it's very dispersed, the particles, but over time, at a certain point, in this case oscillating, it will form these small compartments which will stay for a certain amount of time. And this can be very important, and then after a particular point it disperses. It disperses, there we go. And therefore it enables a very temporal and dynamic feature of the cell which can have huge advantages. And some of the advantages are listed here. And for some of the people who are a bit more familiar with biology, they may recognize some of these functional groups. But I'm just going to focus on one in particular, which is very easy to explain, which is the so-called stress granules. And as this indicates, these are compartments which form under stress. So an analogy to this is probably something like in a village. Let's say we're in a village, and under normal conditions, people are just wandering around and living their lives freely. But if a stress comes in the village, for example, the enemy attacks my village, you want to protect what's most important, right? And let's say, I'm from the UK, and we have a phrase called like, to the king. So you protect the king. The knights will surround the king and protect them from the enemies from around. In a very similar way, the cell forms these stress granules, which will form and protect the most important molecules of the cell. And at a particular time, when the stress is over, they'll disperse. And this allows great flexibility to the cell. It doesn't have to destroy important molecules. It can keep them around and just protect them. And at a certain point, they can disperse, and when the stress is gone, and continue doing their normal jobs. And this video is just an illustration of this. You can see the stress granules forming, and stress goes away, they dissipate. This is another group where they're just tagging the key components of these stress granules. You can see them form and dissipate. And therefore, this isn't over the lifetime of the cell. This is within seconds this occurs over and over again. And this enables great flexibility for the cell. And, these are some of the advantages which these kind of temporal compartments, very dynamic compartments, allow. For example, I talked about D, which is the storage and release associated with stress granules. Another example, if you remember your high school chemistry, is enzymatic reactions. If you're doing a reaction, you want all the components in one place. Therefore, you can put a tent up, stick all the components in one place, and they can interact. It's a bit like thinking, for example, if you have a field and you have a board meeting, let's say, and it's much more advantageous to put all the board members, let's say, in a tent, so they're close together and they can interact and have a debate and discuss what's going to happen. It's not very advantageous to have them in a field where they're shouting from one end of the field trying to discuss whatever. Nothing's going to occur, right? So you want them together, but after a while the board meeting finishes and there's no need for the tent or for this tent to exist anymore. And therefore these kind of temporary, short-term compartments are very important for the cell. However, with every pro, there's obviously a con, and every yin, there's a yang, for example. And what seems to be the case in these cases, it seems to be aging to a certain extent. So when you're young, you're quite sprightly, right? And when you get a bit older, your kind of back starts hurting, you're less enthusiastic about doing stuff, and then this kind of deteriorates as you get older and older and older. And the same seems to happen with these. If I really push this tent analogy too far, it's similar to this. Like when you're young, you kind of put the tents up and down, you have no problems, no issues at all. When you get a bit older, maybe you put the tent up a bit slower, they stick around for a bit longer. When you're much older, you just go, well, I can't be bothered to put that tent down, we're going to leave it there. The same seems to happen with this. So from left to right, these are very young cells and very old cells. And you'll watch basically on the left, they'll disappear very rapidly. And on the right, they disappear slower and slower and slower. Until on the far right, in essence, they seem to stick around for a long period of time and don't dissipate at all. And this is a major problem because it kind of contradicts the whole advantage of these highly dynamic features. Rather than disappearing, appearing, disappearing, appearing, they're just sticking around. And analogy again, tent analogy again, is like, this is a famous festival in the UK, but I mean, this is like people leave the tents around, and this is actually a farm, and this is obviously going to cause huge issues to the farm because rather than just ploughing the field up and down, they're suddenly just going to start trapping all the machinery in the farm with the tents, and this obviously slows it down and slows down the whole processing of the cell. Another example is, for example, if you have a tent here, for example, if you wandered in and you found a tent in the middle of the auditorium, you'd find that it's a little strange, but I mean, you could deal with it, right? But if there's more and more TENS present, they kind of start tangling together and forming these meshes. And these could block, for example, the aisles, but we can always take the other aisle. They could block one door. It's fine because we can use the other door, it's not a problem. But if they start blocking all the doors, then we're in trouble because we can't get out. And it seems to be a similar kind of thing is happening with the cell, where these temporary compartments just don't disappear and start building up and causing so-called aggregates. And this is what seems to be occurring. And obviously these aggregates are closely related to a whole host of neurological diseases, such as, for example, Parkinson's, Alzheimer's, motor neuron disorders. We often see these aggregates forming. And what we are investigating more and more at the moment is the relationship between these highly dynamic, where's my mouse? These highly dynamic physiological granules, which I've tried to talk about the advantages of them, and how they transition into these pathological aggregates, which are highly deleterious for human health. So the relationship between these is what we're interested in exploring in more and more detail. And one of the ways we're trying to do this is in collaboration with some people in UNSW, is to try and create to understand. So we can take some of the features which we know are important for the creation of these granules, and we can make them form under particular conditions. So you can see we actually just shine light and they can form. And by measuring the changes in how these particles form over time with different environments, we can try and develop, for example, we can first try to develop an understanding and then potentially also get drugs to target these features as well. So what I've tried to talk about today is, when we're young, we have very healthy neurons. We have these dynamic assemblies which are very important for a whole host of very important functions. As we get older, they seem to form these aggregates which seem to stick together. And when enough of these aggregates form, they seem to be a link to the disease neurons. And the relationship between these two phenomena and how we control them, I believe, is very important for understanding neurological diseases. Thank you very much for your time.